Excellent. Ah, parfait. Nous sommes online. Okay. Hello, everybody.
There is no. Perhaps we should wait.
You cannot see?
Yes, I can.
But
everybody.
Okay.
Yes.
So we are sorry. We have been facing some technical difficulties here. Somehow it was frozen, and we had to restart and open a new Hangout link. But I hope you're all online now. We are waiting a bit so that more people can join. Now we have already 5. And here we had 9 with the other one. I hope you all find us. Okay. So I'm Hilde, your community manager of the MOOC HIV Science. And today I present to you both MOOC directors, Michaela Müller-Trudwing and Asier Saez-Sirion. And I'm very happy to tell you that now actually 4,424 students and people are watching and attending our MOOC in 131 countries, with a majority in France, Morocco, Tunisia, Cameroon, the US, Algeria, Europe, and South Africa, and many, many more countries. We have covered now all weeks of the MOOC, but you still have 2 more weeks' time to finish the lectures and tests. And we encourage you also to take the final test. And if you want to register for the certificate, and actually the certificate will be online, registration will be available for the certificate from 7th January on. It was a frequently asked question by you. So you can register for the authenticated certificate from 7th to 20th January 2019 with the links that will be provided on the FUN platform. And now I come to the questions you have been posting in the discussion platform that our directors will answer. And I come to the first question, which has been posted by an anonymous person. And it is the following. If HIV controllers are able to maintain a very low viral load that is undetectable and a high CD4 T cell count, how do we test if they are still infected? And I give over and hand over with that answer to Michaela Müller-Trudwing, or no?
Yes. So hello everybody, and sorry for the technical problems that we have. So when we say that HIV controllers have undetectable virus or undetectable viral loads, this means by clinical classic tests, and then the tests that are doing in routine in the clinics. But very often we can detect the virus with more sensitive, ultra-sensitive tests that we use in the laboratories. So for most of the HIV controllers, we can detect the presence of the virus integrated in the cells, doing ultra-sensitive HIV DNA test, and even in some cases, some circulating virus with ultra-sensitive RNA plasma test. What is true is that in some cases, very extreme cases of HIV control, we cannot detect the virus with the techniques and the sample accessibility that we have in the laboratories. And we cannot conclude whether these individuals may have eradicate or control the virus to so low levels that the virus will never come back again.
Okay, thank you very much. So we come to our next question, which has been actually asked by Eric. For him, it was not clear if conventional dendritic cells are infected or not in natural hosts without disease progression, so chimpanzees or African green monkeys. And I hand over with that question to clarify to Michaela Müller-Trudwing.
Hello, everybody. So this is, again, an excellent question. So by conventional dendritic cells, one would think at myeloid dendritic cells. Unfortunately, this is understudied for the natural host, so there are not many data studying whether MDCs, myeloid dendritic cells, are infected. There are data in the literature on whether plasma-situated dendritic cells, so another subset of dendritic cells, are infected in natural host, and in fact they are. The data indicate that plasma-situated dendritic cells are infected as much as plasma-situated dendritic cells in humans. So the infection rate in these cells would not be lower in AGMs or Sutimangabis than in macaques or humans. Regarding conventional dendritic cells, one has to note that it's anyway very difficult to know how much they're infected, because the susceptibility to the cells to the virus depends on many factors, on the maturation state of the cells, on the many different subsets of dendritic cells. So it took years in humans to define whether they're infected or not. So I think this is a huge question and merits many years of investigation. Thank you.
Mm-hmm. Thank you very much. So Eric had also another question. He said, and he asked, in the in vitro co-culture experiments of Olivier Schwarz et al. Regarding the virological synapses, what is the type of dendritic cells used?
So in there are different dendritic cells that were used in the studies. It depends to which paper you refer. Most of the time, when he uses conventional dendritic cells in his studies, we used myeloid dendritic cells that were derived in vitro from monocytes. So the monocytes derived myeloid dendritic cells, or MDCC. In some studies, he also analyzed the capacity of plasma-situated dendritic cells to produce interferon alpha or to sense the virus. And in this case, he often used plasma-situated dendritic cell line, which is called gene22.
Okay, thank you very much. I hope it is clarified now. So we had a question that actually was asked to Virginie Supervi. Unfortunately, she couldn't be here today, but she provided us with the answer. The question is the following. Do you think that the low prevalence in general populations in settings as Latin America is real, or that it is due to the low access to diagnosis tests and low education of the population? The number of pregnant women is high, and I think that this can resemble a low active surge and a false number of the epidemic.
Okay, so we have the answer here by Virginie Supervi, who cannot be here by herself, but she sent us the answer, and I will read it. So apparently these data are absolutely real, and there are several reasons for that. Indeed, in these countries, they don't have low access to HIV testing, and there's no low education. Among pregnant women, HIV testing coverage is high, as mentioned, and nevertheless, higher prevalence remains low. So in Latin America, the vast majority of HIV cases are diagnosed among key populations. Thus, at the national level, the prevalence of HIV remains low in the general population. But it can be high in some key populations, such as men who have sex with men and transgender. We think within the same country, some regions can be more affected than others and have a higher HIV prevalence than at the national level. As an example, Brazil, the southern region appears to be more affected by HIV than the other regions. So yes, the answer is yes, it's real.
Thank you very much. So the next question, which also addressed the lecture of Virginie Supervi, is the following. HIV prevalence is high in key populations all over the globe. How do we target key populations in countries where men having sex with men is illegal? This group of people, like MSM, female sex workers, or intravenous drug users, might be harboring resistant strains that can, if not detected, lead to a different situation to handle. What techniques can be used to target those key populations? So this time, Asier is answering.
So I will also read the answer that was provided by Virginie. Targeting MSM in countries where same-sex sexual activity is illegal remains a complex issue. However, there exist ways to reach and target criminalized and stigmatized populations thanks to the work of NGOs. In addition, a recent study suggested that the distribution of self-tests may be an additional tool to increase HIV testing among MSM. But eventually, the burden of HIV among key populations will not be significantly reduced without a strong political will and key population-friendly policies. Regarding resistant strains, if people do not get tested, they have no access to the treatment, so they will not harbor or transmit resistant viruses.